Okay, welcome back. So you're now in the exon-skipping strategy session. If that's not where you wanted to be, then please find the room that you need to be in. And also because we have breakout sessions and there's not that many people, if the people who are in the room can try to come as much forward as they feel comfortable with, that will make it look more comfortable than with, say, all the gaps in the room. So I'm Annemieke Aartsma-Rus. I've been working on exon skipping for 23 years. And I think what we know is that it works. And that means that exon skipping can restore dystrophin. That's what we know. But we also know that it needs to work better. And by that I mean we need to make it more efficient. We need to get more dystrophin, but also we need to target more exons than the 3 that we have drugs for approved now. So today we have presentations from 8 companies presenting on how to make it better. They all have just under 6 minutes, and they will come one by one, and then at the end there will be a panel session, so they'll be on the stage, then hand over to the next person, and afterwards everyone come back on stage and we can have a panel session. For the presenters, if you're out of time, there's a clock here, so you have no excuse. If you're out of time, I will come up to you and activate the trapdoor if necessary. So not to waste the precious speaking time, I'm going to introduce the first presenter, which is from Sarepta. Please come up to the stage and present the slides.
Hi, everyone. It's a privilege to be here with you all today. My name's Mahima Chojar, and I'm with the medical team at Sarepta Therapeutics. These are forward-looking statements. As many of you know, Sarepta is a public company, and today I'll be covering some of our ongoing clinical research. Sarepta has deep experience in Duchenne and it remains dedicated to the Duchenne community. As you can see, there are several programs we use, or different approaches we use in our programs. Today I'll be covering exon-skipping therapies. Starting with PMO, which is one of the core exon-skipping technologies Sarepta uses, there's currently 3 products that have received accelerated approval in the US. I'll talk a little about that more on the next slide, as well as our late-stage study that's currently enrolling called Mission. These are the 3 products that have received accelerated approval, starting with exon 51 skipping, moving on to exon 53 skipping, and then lastly, exon 45 skipping. These are the late-stage PMO studies in Duchenne. I'll namely focus on the ones that are enrolling, starting with Mission. So this is our high-dose and dose comparison study for exon 51 skipping treatment. It is currently enrolling ambulatory boys between the ages of 4 and 13 years old. We also have our real-world evidence study called Evolve. It's enrolling across all ages and all 3 treatment platforms. A little bit more about Evolve. So this is Sarepta's observational PMO study, and it's designed to collect real-world evidence from those who are receiving one of the 3 therapies I mentioned before. In terms of data, there's no additional visits or special assessments needed, but you would just need to provide consent in terms of getting that information directly from the medical charts. I think we all know the importance of the real-world evidence studies, but just want to reiterate the immense amount of value they provide in terms of safety and clinical outcomes, just to ensure that the drug is behaving the same way in clinic as it did in the clinical trials. Moving on to PPMO, our SRP-5051, which is our investigational next-generation RNA-targeted PPMO therapy. So essentially the way this works is it's it's exon 51 skipping treatment with an additional peptide attached. So the intention behind this is to get more dystrophin expression or production, in addition to a more convenient monthly dosing schedule. Momentum is the ongoing trial for SRP-5051. There's 2 parts, Part A and B. Both have completed enrollment. You can see on the left here a study overview of Part A, which is the multiple ascending dose study. I'll be focusing more on the 20 milligrams and 30 milligrams per kilogram dosing in a couple slides. But this was given IV infusion at a monthly dosing schedule. In terms of eligibility for Momentum, it included ambulatory or non-ambulatory males with DMD amenable to exon 51 skipping in the age range of 7 to 21 years old. Part A of Momentum, so these are the dystrophin expression results. In gold on the left, you'll see the PMO treatment at week 24 compared to Momentum, which is SRP 5051 at the 20 milligram and 30 milligram dosing seen in purple. So comparing those results just even at the week 12 mark to the approved PMO therapy, you can see there's already a significant increase in dystrophin expression. I do want to caveat that with these are very preliminary early results and in a limited data population, so there is more research and clinical development that's warranted. Which is why we have Part B of Momentum, which has also completed enrollment in a similar eligibility criteria in both US and ex-US. And this will be, the intention is for it to be a pivotal trial, so if successful, the plan would be to seek accelerated approval from the US FDA. Also want to touch on our Sarepta Assist patient support program for those interested in exploring approved treatment options with Sarepta. Essentially, you get assigned a one-on-one personalized case manager that helps you through financial logistics, treatment logistics, or other treatment-related needs that are necessary for starting as well as staying on treatment. For those interested in contacting Sarepta Assist, there's a phone number and website listed at the slide below. A lot of our Sarepta colleagues, both in medical and patient affairs, are walking around this week at the conference, so feel free to flag one of us down. You can also reach out to us at advocacy@sarepta.com. Lastly, I just want to thank all the patients and families in our clinical trials. Truly, these therapies would not be possible with their participation. Thank you.
Thank you very much. Also, thank you for perfectly staying on time. So let's see whether the next presenter does as well, which is from Dyne Therapeutics.
Great. Hello there. Thanks. So my name is Ash Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. And it's really a privilege to be here today. Thank you to Pat Furlong and the entire PPMD team for inviting us. It's really humbling to be with this community, and we hope to share some of our data as well as our clinical trial plans quickly. These are forward-looking statements. Please refer to our website if you would like more information. I always like to start with this slide. So at Dyne, it all starts with our mission, which is to develop life-transforming therapies for people with serious muscle diseases, with Duchenne being a priority. This is truly core to who we are and what we strive to do each day. I'm going to spend a couple of minutes on our scientific platform. Probably the most common question I get is, what does your platform do, how does it work, and how is it different from other modalities? So very briefly, Dyne is trying to solve one big challenge, and you're going to hear this theme throughout these presentations: how to more effectively, more efficiently deliver exon-skipping drugs to muscle. And so we've developed something called the FORCE platform. And what you can see, there are three components: the Fab, which is an antigen-binding fragment, a linker, and an antisense oligonucleotide, in this particular case, an exon-skipping PMO. So on the left-hand side, you see this Y-shaped figure. It's a monoclonal antibody. Well, we've taken a portion, the top portion of that antibody, called the Fab, and engineered it to bind very tightly to something called the transferrin receptor 1, or the TFR1. Why did we choose that target? Because it's ubiquitous. It's highly expressed across skeletal muscle, including diaphragm, cardiac muscle, and also smooth muscle. And as I mentioned, the goal is to improve delivery of the exon-skipping PMO. We then connected with this linker. It's a clinically validated linker used in products that are in the market today. And then we attach it to an exon-skipping PMO. So our goal for Dyne 251, which is our investigational medicine, it's designed to enable skipping of exon 51 to make shortened functional dystrophin. This is a snapshot of the extensive preclinical data that we've generated. Before we get into the clinic, there's a lot of work that has to be done to give us confidence that we're going into the clinic with a drug that will potentially be safe and efficacious. We've done experiments in patient cells, as well as in mice models, standard models in Duchenne research, as well as in NHPs, non-human primates, or monkeys, to look at exon skipping, as well as safety and tolerability. All of this data has given us great confidence to move into the clinic. This is just a snapshot of the extensive preclinical data we've collected. And this is in the MDX mouse, which has a mutation in exon 23. So we developed a force conjugate to target exon 23. And really, the bottom line here is that we were really pleased to see these levels of dystrophin restoration in key muscles like quadriceps, diaphragm, and heart. And especially in the diaphragm and heart, where other modalities have had a lot of difficulties to be able to penetrate. So we are undertaking our phase 1/2 clinical trial to evaluate Dyne 251 in patients with Duchenne. Very importantly, before we finished the design of the trial, we had input from advocacy groups such as PPMD, the Duchenne Community Advisory Board, which comprises of advocacy leaders from around the world, as well as thought leaders, neurologists, pediatric neurologists, adult neurologists, physical therapists, pain medicine. So we've really tried to incorporate all of this thinking globally from thought leaders into the design of our trial. And what you can see here is our population consists of patients with DMD that have mutations amenable to exon 51 skipping. We have a pretty expansive age range, 4 to 16 years of age, that include both ambulant and non-ambulant participants. Our primary endpoints are safety and tolerability and dystrophin restoration, and we're looking at a number of key secondary endpoints. We're really trying to understand how the drug enters the body and exits the body. We're trying to understand the level of exon skipping and other measures of dystrophin, and of course looking at multiple assessments of muscle strength and function, measures that you know, like the NSAA, time function tests, upper limb function, cardiac function, respiratory function, and we're also trying to look at novel endpoints like stride velocity, which helps us complement the current measures that we have in this study. We have 3 periods, essentially, or 3 stages. We have a placebo-controlled, multiple ascending dose study of 24 weeks, period of 24 weeks, followed by an open-label extension of 24 weeks where every participant is guaranteed to be on drug, and then a long-term extension of 96 weeks. And we're really pleased to say that we will have initial safety, tolerability, and dystrophin data in the second half of this year. And this slide just shows some of the core inclusion and exclusion criteria. You can go to our ct.gov page to get the full set of criteria, as well as all of the sites around the world, and certainly in the US. And we continue to open up sites in the US. And I just want to say thank you again. Thank you for the sacrifices you've made for decades. None of this would be possible without the families and all you do for the community. So thank you.
Thank you very much, Ash. Next up is PepGen.
Hi. Thank you very much to PPMD for the opportunity to speak today. It's always great to be at a PPMD meeting. I've actually lost count of how many of these meetings I've been at in various capacities. I'm here to tell you a little bit about PGN EDO51, our exon 51 skipping therapy for Duchenne muscular dystrophy.
There's a clicker.
Next slide, please. There we go. There are our disclosures. Again, if you have trouble thank you. So if you have trouble sleeping, you can read our disclosures. This slide really just shows you what it is that's special about PGN EDO51. As Ash already described to you, one of the big problems with oligonucleotides and exon-skipping drugs in general is delivery. We know they work. If they can get into the cell, specifically into the nucleus, they really work quite well, but the problem with the first-generation drugs is they didn't penetrate well. Our technology is a small peptide, or a little piece of a protein, that we attach to the oligonucleotide and allows it to penetrate into the cell. So if you think of it as you have the most important piece of information in a Zoom meeting, but you can't connect. Our peptide allows you to connect, get through that error message, get into the room, and share that really important piece of information. In this case, skip over exon 51 and produce dystrophin. And that's what our technology does. This is our pipeline. I'm talking today mostly about exon 51 skipping. We also have programs in looking at skipping of a number of other exons at earlier stages in development. And we're learning a lot from our exon 51 program that will allow us to move more quickly with the other exons as we move forwards. We also have a program in myotonic dystrophy type 1. We are a neuromuscular disease company really working to solve the problems of neuromuscular diseases. And a lot of that comes down to that peptide. We get into various tissues very well. We have great transduction of skeletal muscle, including the diaphragm, cardiac muscle, that's the heart, smooth muscle, that's the gut, and we even get a small amount into the central nervous system. So we get everywhere that matters for people with Duchenne. I'm going to summarize a lot of data very quickly because five or 6 minutes is not very long. Basically, we have looked at EDO51, or the mouse equivalent, EDO23, in mice, wild-type mice, MDX mice. We've looked in monkeys, and we've done a phase 1 study. And the summary overall is we see great levels of exon skipping and really great levels of dystrophin production in the MDX mouse and every model we've looked at. But I'm mostly going to talk about our phase 1 study. Why did we do a phase 1 study? We looked in healthy volunteers. Why? Because we wanted to understand our drug as best we could. We did a study in healthy adult males. We had 32 men come in. They spent 11 days in the clinic. I wouldn't want to ask you or your sons to do that. But we could bring these guys in. We studied them intensively. We looked at every safety marker we could imagine. We took biopsies on day 10 and day 28, so we could learn as much as we can about our drug. So when we go into boys and young men, we'll know as much as we can about it. What dose is likely to be effective? How often are we likely to need to dose? And how safe is it? So I'm going to tell you a little bit about this study. We had 8 people in each cohort. We went from 1 mg per kg to 15 mg per kg. And as I mentioned, we did some biopsies and a lot of safety assessments. This summarizes what we learned. In terms of target engagement, exon skipping, we saw the highest level of exon 51 skipping that's been observed after a single dose. These were healthy volunteers. We only did a single dose, but we saw really nice amounts of exon skipping. In terms of pharmacokinetics, we saw a really high level of oligo delivery after a single dose, and we were generally well tolerated. So all of that really supported us moving forwards. This is just a summary of huge amounts of work in one simple slide because, again, time is short. On the left, you'll see our 10 mg per kg cohort. This was a dose where we saw no adverse events that were more than mild. And we saw 1 to 1.5% exon skipping by ddPCR. If you look at our monkey experiments going back, when we did a single dose at 20 mg per kg there, we saw 2.5% exon skipping by the same assay. When we repeated that assay, we repeated that dose, so 4 doses in a monkey. We multiplied that 2.5% up to nearly 35%, so 14 times the amount of exon skipping. So this really encourages us that with repeat dosing in boys and young men with Duchenne, we'll see meaningful amounts of exon skipping and therefore meaningful amounts of dystrophin. The other thing I want to point out, from day 10 to day 28 in that healthy volunteer study, we saw similar amounts of exon skipping. That suggests that if we dose every month, and this is an IV infusion that we would be giving every month, we will see accumulation over time. We don't need to dose more frequently. Exon skipping and presumably, therefore, dystrophin production will be supported over that time. So that really supports the fact that we want to move forwards into trials with boys and young men. And we're launching 2 trials. Connect 1, we've just received our approval letter to start in Canada. This is an open label MAD study, which will be starting soon. And Connect 2, our global randomized double-blind placebo-controlled study, will be starting a little bit later, which is a global study. So overall, in conclusion, our data is a really strong package to support moving forwards in people. We're really excited to be starting clinical trials, and I'm happy to answer questions now or at our booth later on. Thank you.
Thank you very much. Next up is BioMarin. And after BioMarin comes Wave, so that you can be prepared.
Good morning. My name is David Neal. I am a physician. I sit within the clinical development organization at BioMarin. It's a real pleasure to be with you today to talk about some of BioMarin's research into Duchenne muscular dystrophy and some of the plans we have for the future. I'm going to cover our research with regard to another exon 51 skipper, results from some of the preclinical studies we performed to investigate the efficacy and safety, and then, if I get time, hopefully cover some of the future clinical trial plans. I do want to point out BioMarin's been around for about 25 years at this point, and then, as of now, patients remain very much at the heart of everything that we do, and our scientists embrace cutting-edge science and technology to try and deliver meaningful medicines to treat patients living with rare genetic diseases. Obviously, I have to cover the forward-looking statements here, as have my peers. So today I'm going to talk about our research for the possible treatment for DMD. As I mentioned, 25 years ago is a long time, but in 2014, BioMarin acquired a company which at that time was in phase 3 clinical trials for another ASO, drisapersen. Unfortunately, in the phase 3 trials, they were not positive, but we didn't stop. We continued our research in the background to develop a new antisense oligonucleotide. And I'm pleased to say that we're now at a point where we're discussing with regulators as well as the DMD community at large our results from our efficacy data in the non-clinical studies as well as potential to move to trials. So what is this I'm about to talk about? BMN 351, it doesn't have a name yet. It is still an investigational drug, so it's not yet approved for human use. However, in comparison to drisapersen, we looked very much at chemical modifications we could make to improve stability, and we also identified a completely novel target binding site to hopefully improve exon skipping potency. It will be administered, in contrast to drisapersen, intravenously rather than subcutaneously. So the main research questions we asked in our preclinical studies were regard to percentage of exon 51 skipping. We also looked to see if there was more dystrophin produced as a result, and we also looked at functional ability in those animals too. So as you will be aware, in DMD there are standard mouse models which are modified to actually express the DMD gene, and we've studied this actually across 20 non-clinical studies, but there's two I'll talk about today. One was using a screening compound where we dosed for 12 weeks, and we looked at the exon skipping there, and then the other we dosed for 5 months in these mice. So this first 12-week study, you can see here for reference, drisapersen, only about 1% of exon skipping seen in this particular model. The oligos that are listed, A through J, are molecules where we only made some chemical modifications to try and improve stability and immunogenicity. But you can see here with the pink line at the end there, we're seeing this is with the new target site, and it seems to increase potency with regard to exon skipping significantly. We move forward to, this time it's in an MDX mouse model, and we dosed again, this time for 25 weeks, and you can see the results here when we looked for dystrophin expression either 1 month or 3 months following the last dose. You can see here in firstly thigh muscles, this is expressed as a percentage of the wild type situation. We're seeing anywhere between about 30 and 50%. This is dystrophin production, and that maintained 3 months after that dose, so it continues to accumulate and be produced. We're also seeing penetration and dystrophin production in the heart muscle as well. Again, slightly to a lesser extent than skeletal muscle, but this is potentially indicative of the potential for benefit maybe in later stage disease once we get to clinical trials. So in the same 5-month, 25-week dosing, we also looked at functional ability in those animals, and this is using a motor rater score. Here, a higher score is worsening of the animal's ability to move. And you can see here in the placebo arm, the no treatment, that's the dark purple line at the top. The animals do indeed worsen over the course of the study. But with both doses we studied, you can see an attenuation of that progression, so the animals don't get any worse. In fact, they seem to trend towards improvement in terms of their ability to move. Running out of time here, so I'll be quick. Obviously, safety is at the core of everything we look at. We know that antisense oligonucleotides have a well-known class effects, and we've studied safety, of course, in our animals, and we're seeing similar safety effects, but nothing that would preclude moving on to clinical trials. So we'll be looking for those safety features in the clinical trials. So this is forward-looking, and we hope to be able to initiate clinical trials in the near future. These will be in ambulant patients who are amenable to exon 51 skipping, aged between 4 and 10, and have not previously had a gene therapy. So any further questions on the trial or anything else I have to say, please contact at the email address here or find myself in the lobby later. Thank you for the time.
Okay, thank you very much. So next up is WAVE, and after that will be Entrada.
Never want to rush anybody off, so thank you. And good morning, or good afternoon, and thank you for giving us the opportunity to speak with you today and for the conversations that we've been having over the last couple of days with many of you and thinking about our wants, needs, and really hope for the treatment of DMD. I'll share some requisite forward-looking statements, but the most important thing, thank you, is really the opportunity that when we had some of these conversations this morning, one of the things we heard a lot of is, we're happy you're back in DMD. And I really reflected on that because it is important to know that WAVE isn't back in DMD, we never left. The DMD community has been important to us for a number of years. I've been the CEO of WAVE now for 10 years. And this community has contributed so much to helping us advance and think about our future pipeline. So when we made the decision and we thought about going back into DMD, some of you may recall that our first exon-skipping therapy didn't have positive data. We didn't see dystrophin production. And we reflected and spent a lot of time with the community, a lot of time with our team, really thinking about why that is, and went back and really revisited chemistry. You heard a lot about different chemistries. We really had two main purposes in thinking about our exon-skipping therapies going forward. One, we wanted to improve skipping efficiency. As you heard today, the goal is about getting as much dystrophin production as possible. But two, we wanted to do that without putting anything additionally onto the molecule in terms of conjugates. We wanted to do that, maximize skipping without adding potential liabilities to safety. And so we were able to demonstrate, and we'll walk through our process, and I'm not going to give a chemistry lecture on our improvements, but our improvement was really adding a neutral charge to the backbone. Now, we also heard a lot about different models, and one of our reflections from our first study was a lot of work in DMD is done in the MDX model. And we really reflected on translation and spent time thinking about a model called the double knockout model. So this is a model that doesn't have dystrophin, doesn't have utrophin, and so therefore, these mice progress as if they have no dystrophin. Therefore, any benefit that's seen in this mouse is really a reflection of restoring dystrophin levels. And you can measure survival, you can measure respiratory functions in these animal models. In light blue, actually in orange, is the survival of control. So these mice have a rapid mortality, they die at about 8 weeks. And what we saw is when we used our first generation backbone, so similar to our first compound, at a dose that was about 10 milligrams per kilogram, so double the dose that was in the clinic, we do see a slight survival advantage. But what was exciting for us is with the new chemistry, at a dose of a human equivalent about 10 milligrams per kilogram, we saw 100% survival in these mice. That means that we were restoring function and restoring dystrophin to a level that was allowing these mice to progress. What's important, given our past experience, is we then cut that dose in half, so a human equivalent dose of 5 milligrams per kilogram, we gave it every other week, and we still saw that we were at the top end of that therapeutic index, meaning we were still producing dystrophin substantially in these mice. Now, beyond survival, we could actually study function. So what we saw is with this PN chemistry modification, we were getting to heart, we were getting to diaphragm, and if we actually looked at respiratory function in these mice, we were restoring that back to wild-type levels, meaning this was back to a normal phenotype. Now, as we think about our preclinical data moving into our clinical study to give us the confidence to come back, we were able to look at concentrations of drug in various tissues. And what we saw was we actually had 5 times the amount of drug from the skeletal muscle in the diaphragm. And impressively, we saw 25 times the concentration of drug in the heart. So therefore, the chemistry adaptations that we've been making, without having to put delivery conjugates on, we're delivering a functional medicine into these tissues. If we think about dystrophin levels and think about exposure, we saw 74% dystrophin at a 10 micromolar concentration. So this gave us the confidence to come back into the clinic, but do so responsibly. So we did a small study to really answer the question of, do we see a difference with our PN conjugated drug, so N531, compared to our first generation drug? We did this study in an initial cohort of 3 boys. We dose escalated within those cohorts, and then at the end did a very small study. So this was 3 doses over 6 weeks, so an early time point to really answer 3 important questions. Do we see drug in the muscle? Do we see exon skipping? And is it safe? And because we had the muscle tissue, we'd also look at dystrophin levels, again, at a very short time where we wouldn't anticipate seeing protein. What we saw in our clinical trial in these 3 boys was very high muscle concentrations of drug, 42 micrograms per gram. If you think back, higher than what we saw in the non-human primate. We saw the most amount of exon skipping that's been seen in a clinical trial to date at a 6-week time point, which is 53% exon skipping. And we were below the level of quantification for the dystrophin protein at this early time point, with a half-life now of a drug that's 25 days. So as we talk about the ability to dose less frequently, we do believe that this is a monthly potential dose. I'll go really quickly.
No, you have to wrap up.
I'll wrap up. So, nicely done. So I think what I'll say in final words is safe and tolerable. We saw the same profile as you'd see with PMOs. No evidence of oligotoxicities. And equally important, we're now initiating the potentially registrational phase 2 study where we'll expand that population up to 10 boys and run that study over a period of looking at muscle biopsies at 24 weeks and 48 weeks. Thank you.
Okay, thank you. And we have the next session and try to be on time in that session. Next up is Entrada, and after that is Avidity.
Okay, we know she is not kidding about the time. Hi, everyone. It's my pleasure to be here and represent Entrada Therapeutics. I'm Narissa Crea, the Chief Medical Officer. This is our forward-looking statements. We are a public company like many others you've seen today. So I want to start with our commitment to the Duchenne community. Are there families in the room that are interested in exon 44 skipping? If so, if you could just raise your hand. Great, thank you. One of the examples of our relationship commitment to the community is listening to the community. And our decision to go into exon 44 as our first program is a great example. We listened to the patient advocacy groups, the families, the thought leaders, and made the decision that we would start with exon 44. Are there families that are interested in exon 45? Okay, same thing. So thinking about how are we staging the programs that we're focused on. We have a program for 44 skipping, 45 is following that, and then we have discovery efforts in exon 50 as well as exon 51. So one, I just want to say we appreciate the community, we appreciate your voice, and we're committed to listening. So you've had opportunities here from several other companies already, and you've heard a very common theme. We're all working to try to get better exon skipping and better dystrophin production. And each company has a different approach to doing this. Our approach we call endosomal escape vehicles, or EEVs. And it's important not to confuse that with AAV related to gene therapy, so completely different. Our EEV platform, those EEVs are actually small pieces of proteins that are cyclical, and that's one of the differentiating factors compared to some of the other programs you've heard about today that have linear peptides. So we're cyclical, and what that leads to is a differentiation from the standpoint of not only getting more of the drug into the cell, but getting it specifically to where it can have action, and that's the nucleus. For a 44 program, because of the limited time, I'm going to share one data slide with you and try to make a couple of points about our data. On the left-hand side, you can see exon skipping in a non-human primate. And in this, after 7 days, you can see that we see significant exon skipping across multiple skeletal muscles, but importantly, the heart, and also an important respiratory muscle, the diaphragm. On the right-hand side, something that we are excited about from the standpoint of the way that we will be delivering this therapy is that we see significantly prolonged exon skipping after a single dose. And so in that, we are looking at developing our therapy to be an every 6-week therapy, and there's a possibility that it could be delivered in intervals even longer than that. It is delivered by intravenous approach, so similar to others, but the big differentiator here that we're really excited about is that time interval between when we think the drug will need to be delivered. So in summary, we have very effective exon skipping with our 60144 program. We see robust dystrophin production in patient cells. I didn't show that data today. It's available on our website and our corporate presentation, and that prolonged duration that I was just speaking about. I'm going to move quickly to our 60145 program. Again, this is for boys that are amenable to 45 skipping. And here in this data slide, we're actually looking at patient cells, so experiments we've done with patient cells. And on the left-hand side, you can see exon skipping, as well as dystrophin production. And again, just like our 44 data, we get very significant exon skipping as well as dystrophin production. On the right-hand side, I stress that we're seeing a lot of increase in the cardiac muscle, and we're very excited about that. So on the right-hand side, you're seeing dystrophin production in cardiac muscle cell from a patient with Duchenne muscular dystrophy. So in summary, we're very excited to be in the Duchenne community. We look forward to many years of working with this community. Our 44 program, we look forward to moving into the clinic near term. Many of you will know we are on clinical hold with that program, but we're committed to resolving that and moving that program forward. Our Exon 45 program is slated to be in the clinic sometime next year. And then discovery efforts underway again for Exon 50 as well as 51. And our team from the Boston area says hello, and again, thank you so much.
Thank you very much. So next up is Avidity, and then last is NS Pharma.
All right. Well, thank you to PPMD for the invitation. It's great to be back here with all of you. My name is Mark Stahl. I am a neurologist, and I've taken care of folks who have Duchenne. And I work at Avidity Biosciences, where we are working to make new medicines for folks who have neuromuscular diseases and other rare diseases that we can treat with RNA therapeutics. These are our forward-looking statements, as you've seen from other folks. We are developing a technology that also is a novel way of delivering oligonucleotide therapeutics to cells. And in our case, the difference is that we are using a full-length monoclonal antibody that we link up to an oligonucleotide. We call this the AOC, the antibody oligonucleotide conjugate. And while this has a lot of similarities to some of the other approaches that you've heard, it has a few other advantages as well. And so one is that, again, we target a protein that's on the surface of muscle cells called the transferrin 1 receptor. But we also use an antibody that persists in the body for several days, and which is a longer time than some of the other ways of delivering these oligonucleotides. And that is a piece of the technology that enables a longer dosing interval. We'll be testing, as I'll talk in a moment, in our studies, we'll be testing both every 6-week and every 8-week dosing intervals. And of course, the goal of the delivery of our exon-skipping oligo to muscle is to treat the underlying problem of lack of dystrophin. So I'm going to jump into our first study in Duchenne patients, which is called Explore-44. That's named after the fact that we are treating folks who are amenable to skipping exon 44. This study was designed, as I hope many of you in this room know, in conjunction with the real experts in this disease, which really is you. And of course, leaders in the field, expert physicians, expert scientists, but it's really the advocates and the patients themselves and the caregivers who really give us the most important insights into what they need. And we take that information very seriously. And we thank a lot of you who are here today and will be watching our presentations for giving us your input directly. Of course, the primary goal of our first study is to evaluate safety and tolerability of the drug. And in the first stage of the study, we'll be giving drug to 75% of the participants with DMD and placebo to the rest. But after just 3 doses, we'll be moving into a phase where everyone gets treated. Of course, our drug, just like many of the ones that you've heard about, is an intravenous infusion, and it will be testing these 2 different dose intervals that I mentioned. I'm going to just talk a little bit about sort of the important things to get into the study for participants with Duchenne. Of course, you have to have the exon deletion that will make you amenable to having exon 44 skipped. We have a broad age range from 7 to 27 years, a minimum weight. People can be taking steroids or not, and we can accommodate both ambulatory and non-ambulatory patients. There's a few standard tests of respiratory function, cardiac function, and some of the muscle function tests as well to get into the study. This initial study is about 8 months in length, which includes the screening periods and all of the follow-up. But then we expect after the 3 doses in this study that everyone will move over into an extension phase where, again, everyone will receive drug. The study as designed, I want to share a few things with you. It starts off in healthy volunteers, and we do this to avoid giving any doses that we don't expect to be effective in patients, and do all of the safety studies and things like that we can in healthy volunteers before we move over into doses that we really do expect to be efficacious or helpful for the patients. And there are 3 doses that we'll be testing and 2 dosing intervals. One other important point about our study is that we took the advice of a lot of the folks who are talking to us about having the experience of Duchenne to try to really make the participation in the study as convenient as it can possibly be. So there's home health visits, study travel concierge who is involved in setting all of the logistics and reimbursement and things like that for expenses. A couple of just very brief key takeaways that really the Explore-44 study that I mentioned has already begun. We are partway through the healthy volunteer portion, and so we expect to be enrolling patients a little bit later on this year. And through the study, you'll be assigned to receive IV dosing every 6 weeks or every 8 weeks in interval. And so thank you so much to everyone who is participating in our focus groups and talking with us and helping us out. Everyone at Avidity thanks you, and please don't hesitate to reach out to us at our booth, or if you see me in the hallway, or send an email to our email address. So thanks so much.
Thank you. So the last presenter is from NS Pharma, and after that talk, all speakers are asked to come back to the podium, so be prepared for that.
Hey folks, so quick disclaimer, I'm not Dr. Robert Crozier, but I am happy to be here. My name is Jay, I'm a medical liaison, I'm based out of Houston, Texas. And today we're excited to talk about our exon 44 skipping investigational therapy. Forward-looking statements. Quick overview of the presentation. We're happy to say we do have a generic name now, which is brogadierson. And just 2 things, I'll be discussing the phase 1 data, and then sharing some trial information for our phase 2 trial we're hoping to bring to the US. So once again, I mentioned our phase 1 trial. This was conducted in Japan by Komaki et al. And actually, originally, this data was presented at World Muscle Society last year in a poster format. So let's take a quick glance at the phase 1 study, who was included. It's a story of 6 boys. We have 3 in the 40 and 3 in the 80 group. Age range is from 4 to 13. Average, you can see, is about 9 years old. As far as exon deletion, we actually had, out of the 6 patients, 5 had a single 45 deletion, and 1 had a 45 to 54. And all boys were on steroids except for 1. Well, let's look at the results at 24 weeks. So there's 3 colors I want you to focus on. You see the gray bars, that's the dystrophin at baseline, and then you'll see the blue bars, and that's after treatment. We can start on the left. In the 40 group, you can see 3 boys. Their baseline was 6.4 and around 16.6% after 24 weeks. In the 80, you see 8.7 starting, and then up to 24.5. And the third graph, if you just take the average of all those numbers, you can see an average of 7.5 starting and then 20.6. And the yellow color is just the difference between those 2 numbers. And we're happy to say, with only six boys, we're able to find a statistically significant difference between that. As far as safety, we didn't see any drug-related serious adverse events, and all boys actually continued into the extension. So we're very excited with that data there in Japan, and we're excited to announce our new exon 44 skipping trial that we're bringing to the US. This is going to be a phase 2, multiple dose, 2-part study. If we just take a quick glance at it, we can see that we're going to be recruiting 20 ambulant boys from age 4 to 14. It is a PMO, so we have a once-weekly IV infusion. They have to be on a stable dose of steroids for at least 3 months. And as I mentioned, part 1 and part 2. Part 1, just to simply explain it, we're going to be looking at 3 different doses, a 40, a 60, and a 80, each 4 weeks, so that equals 12 weeks. And then in part 2, those 6 from the first part will roll over, and we'll have 14 new patients, so total of 20 patients. From an exon skipping trial, some standard procedures you can expect include muscle biopsies, blood draws, urine collection, physical exam. It says MRI testing up there, and I just want to clarify, this is not cardiac MRI. This is skeletal muscle MRI testing. And you can expect some standard motor function tests as well. For the full list of inclusion and exclusion, you can go to clinicaltrials.gov, but just 2 key exclusion criteria, since there's going to be several questions around this. If you received any gene therapy, unfortunately, you would not be a candidate for this trial. If you have received an investigational drug, you may be excluded if it's within 3 months. So I explained here the exon 44, but our commitment doesn't stop there. We do have an approved product, which is Viltepso, which targets exon 53. And I'll actually be giving that talk in the next room here in a few minutes. And we're hoping to start 50 this year, and we're really working to capture as many patients as we can. And all this isn't possible without the DMD community, the doctors, the patients, the advocates. So I just want to say a quick thank you, as everyone else here up here has done that.
Thank you very much. Perfect, on time. So if all the presenters can come back on the stage, we have 2 handheld microphones, and if people want to ask questions, then they can come to the middle microphone over there. And well, I see the first question is there, but people are still coming up. I'll go on the side. So there's a microphone there. You can stand, you can sit. There's not enough seating for everyone. So Carrie, ask your question.
Given that we need dystrophin not only for mature myofiber health, but also for satellite cell fading and regeneration, do any of you have any insight as to whether your oligos target satellite cells, or are they predicted to, or do you have data showing that they might?
No. Try now.
How's that?
Yes.
All right. Yes, so I guess the question was for everyone, is there data demonstrating satellite cell delivery? One of the studies that we have looked at, so with the modifications we made to our oligos is, yes, they do get to satellite cells, and we have seen that possibility for regenerative muscle.
And are they equally effective as they are in the mature myofiber, or is it more difficult?
So this is early. We'll be providing additional updates on really distribution, but it's a great question and something that we spend a lot of time looking at.
Anyone else have anything to add to that? No.
Thank you.
Thank you. Please.
Hi. Excuse me. First, just a heartfelt thank you for your work and attention to DMD. Curious if you have any insight or if there's any chatter in your industry about creating treatments for the more rare deletions. So, for example, my two boys are 3 through 7, and they're amenable to exon 8 skipping. So curious on your thoughts.
Yeah. So maybe just start with you, and then, because they're probably interested to hear this from all the different companies. So we start on the left, yeah?
Yeah, sure. So as far as perspective from NS Pharma, when I presented the pipeline at the end, and there was percentages that showed, which exon skipping and what the percentage population is there. I think the challenge is with those rare mutations is starting a clinical trial and getting everyone on board for like an N of 1 study. So right now, the goal is, all these new technologies, our goal is trying to prove the efficacy and safety in the broader population before we consider the rare mutations.
Yeah, I think that was well said, and I think one thing that we could maybe add to that is the idea that as we get into more smaller and smaller populations, right, it becomes more challenging to conduct a clinical trial that's kind of a conventional design, right, where you have control populations and you do a study that shows statistical significance on an endpoint. I mean, at some level, right, we will run into needing to recruit the entire population of folks who have a given mutation, right, and so what I think we need to start thinking about is ways that we will translate the data that we generate in these more common exons, right, to maybe a more general framework that maybe regulatory bodies could consider, or something along those lines. That's a bit of a new approach that will allow us to address some of these smaller populations. Right now, that framework doesn't really exist, and I think that's something that we can work together in many ways to help to develop. So I think that's a way to kind of conceive of how that might happen in the future. But I can't really comment on any specific plans that we have as a company. But I think that in order to do that, the general thought is that we'll have to change how we approach things. So.
Yeah, not much more to add. I think I'd be surprised if all of the companies here haven't thought about how to tackle those rare mutations. I'm sure there have been conversations with regulators as well, and I think Mark said it really nicely. And maybe I'll just go a little deeper. Hopefully, we can generate a critical mass of data from these different modalities to demonstrate efficacy and safety, and then leverage that data for the more rare mutations, maybe even to get to a point where you don't need to do clinical trials because it is just not feasible. And so you go in with non-clinical data and you've leveraged the platform. And so I think those conversations with regulators should be happening, need to happen, FDA, Europe as well. And then I think there are opportunities to collaborate with the community and with one another on this.
And I think we had this discussion yesterday as well, like once you know that you need X% of dystrophin to get functional benefit, then well, you don't really need to do the trial, you just need to show X% exactly of dystrophin. Do you have anything to add? Yeah.
Because we talked a lot about opportunity. I mean, I think one of the things that we had done at WAVE prior, so if we go back to the Distance 51 study, is we did have regulatory approval for that study that we were running, and that was a basket study. So we used an augmented placebo design that was really in partnership with the agency to do exactly that, to think about how we can use fewer patients in a placebo arm, be able to use natural history studies that are statistically tested against the clinical trial placebo patient population so that we could run smaller, more focused studies looking at outcome. And one of the extrapolation from that, and we do have the ability to look at same chemistry on top of different exons, so being able to think about running more basket-like designs with studies that we know that the regulators are open to. So I think the opportunity set does exist.
Yes, please.
Just a quick question for Sarepta. In 2016, Exon Dis 51, I believe, got conditional approval pending a confirmatory trial. And I was just wondering where the results for that trial are.
Great question. So Mission, which is our high dose and dose comparison study, is also going to function as that confirmatory trial. So it is currently enrolling. It's been a little bit challenging to enroll it just with the other therapies in our pipeline. So that's been a challenge, but yes, it is currently enrolling.
From 7 years ago? Like if it was 2016, it's taken 7 years to enroll the trial? Yes?
I'm sorry, I don't understand the question.
I'm sorry, I thought 26, so you're still enrolling the confirmatory trial from 2016?
Yeah, there were some delays with Mission, but yes,
Okay.
we are still enrolling.
Okay. And then maybe a last, do you have a question? Yeah. Okay, I was going to ask the last question, but audience comes first. No worries.
Yes, so I'm super excited to see all of the 44 skipping. That's what we've been waiting for and watching it in preclinical for a long time. So if I were to open a spreadsheet and make a matrix that helped me to decide which one I should enroll in, what are all of the columns that I should have to evaluate which trial we would want to choose?
You want me to take this? So we wrote a paper about this very problem, but then from the 51 perspective. So maybe have a look at the paper. It's open access, and it discusses things like, well, what do you know preclinically? Because what we've seen, some have higher efficiency than other, but also about expected safety. What do we know about these compounds? How often will they be injected? And there's no right or wrong question, but you have to see what is for me the right question. So some families may say, well, it's perfectly fine to come to the hospital weekly because we know that this compound is really very safe. And others may say, well, we prefer to have something where there's slightly more risks because it's the first time it goes into a human, but we don't have to come weekly, we have to go every 6 weeks. So there's lots of things to check, but what is important, that's of course based on your own situation. Yeah.
Yeah, and I mean, I would add to that, as it is an individual decision, but guidance with your treating physicians, I think, is one of the critical pieces, because the physicians will have information on the various trials and having those conversations. But, you know, safety, obviously, we're all focused on safety first, efficacy, and then some of the other concepts. That
number of biopsies, there's lots of different things. So I see the clock says 15 minutes, but I know that we also need time to transition to a different room. So I was instructed that we had to wrap up at 1:45, which is now. So what I want to do is first to ask for a round of applause for the tech people, because they had a big job with the 8 different talks and all the different slides. So please, an applause for the tech people. And then also a round of applause for the presenters and the audience. And then that does give you some time to find the next session that you want to be. It gives us some time to find the room that we need to be in next. So I think we are next in Lone Star A3. So if you want to see this session again, Lone Star A3, otherwise find another room. And thank you very much.